So the next unit is on proteins. Proteins are basically these large chain of amino acids that do specific tasks. So the smallest component of a protein is an amino acid. We bring in amino acids from all the foods that we eat that have proteins, and we take those amino acids and construct whatever our body needs. So you bring in protein from as one thing. So let's say you eat chicken breast or an egg or beans, and basically what happens is you take those proteins, break them down to the smallest component, amino acids, and build, here's just a mini list, but it could go on for a really long time, but let's do just structure, the structure of a cell, structure of our body. Enzymes. Enzymes are a specialized protein that do some work. Transport, moving things from one area of the cell or one area of our body to another. Immunity, we have a whole bunch of chemicals that we build, which are proteins, that are designed to kill parasites and bacteria and all these other things. Motion, we have a bunch of cells in our trachea and our nose and even in the back of our mouth that have these little hairs on them, and they're basically always moving to catch foreign things that we breathe in and then basically kick them out. So either kick them out as a sneeze or a cough, or basically just drop it in our stomach to kill them so it doesn't take over our lungs. And a lot of the things that float in the air are fungal particles, and they would love to grow in our lungs, but these little hairs basically kick them out and destroy them. Motion, that would be part of motion as well, and then a lot more. Amino acids, the smallest building block of a protein. So you take a bunch of these amino acids, put them in the right way, they become one type of protein or another type of protein. There's instructions on how to build these proteins, and that's what we call DNA, which we're going to talk about in a second. There's 5 main parts of the amino acid. There's an amine group, there's a carboxyl group, there's a centralized carbon, a hydrogen, and an R group. And this means just that in chemistry you'll hear about these R groups. It means this is the only part that could be variable. And that makes the difference between one amino acid and another. But everything else is a pretty common structure of every amino acid. These kind of components I would never test you on, but you might hear about them again in chemistry. So that's why I gave you these details. But it is time to start stringing these amino acids together. We basically take an OH from one group and an H from the other and get rid of it. And we can make these strings longer and longer and longer. There's about 20 different amino acids, and the R group that we saw before, you can see here they're highlighted in this yellowy orange color, and you can see that they all are a little different. And some are going to be this thing called hydrophobic, meaning they don't like water, and some are going to be hydrophilic, they like water. And because of those charges and those sort of reactions that they have, when they come together and they start to make these large chains, they start to fold in a specific way. So wherever we put these are really going to dictate the shape of what we're going to be building. So as we're putting these amino acids together, what we've done is, for instance, over here we have an OH and an H. We take those out with a dehydration reaction. So hydrogen and OH, we basically made water, so we removed it. So now what's happened is amino acid 1, amino acid 2 have come together to become 2 amino acids, and that bond that holds them together is called a peptide bond. And that peptide bond is just specific to proteins. We can do this with many proteins, and there's called dipeptides and tripeptides, but realistically every protein that we're going to deal with is going to be thousands, if not millions, of amino acids long. So we're just going to go ahead and stick with the term polypeptide. Many peptide bonds holding this protein together, or many amino acids being put together. So you can see a lot of these reactions occur. So let me go ahead and just kind of reshape how you think a little bit, I hope. But when you eat a protein, you're eating a large chain of this. So let's say thousands of them. And your body really can't do much with it until we start to break them down. So what we do is we break this peptide bond, we add a hydrogen to one side and OH to the other, and then what happens is we're going to do this reaction in opposite. So this gets into our stomach, in our small intestines, we're going to do the opposite reaction of dehydration. Do you remember what that one's called? Where we add the water to it? Hopefully you said hydrolysis. If you didn't, that's okay. It's a tough word. And you could just review a couple of the first few slides for this chapter, but you'll see that we added an H and an OH, and that broke these two in half, and that hydrolysis reaction is occurring in your small intestines. It dumps all these amino acids into your bloodstream, and your cells pick them up, and they want to build proteins. So what they do is they put all the amino acids in the right combination, and they remove the OH and H, and they do this thing called that dehydration reaction and make the amino acid chain in whatever shape they need. Do they need an enzyme? Do they need something for motility? They're going to build it based off the instruction manual. Proteins can be broken down or changed 3 different ways. And when they lose their functionality, we call them denatured. So the nature would be them doing what they need to do. Denaturing them means changing them so they're completely lost the function. We could do this 3 ways. We could change it with pH, we could change it with temperature, and we could do it with salinity, salt. And these 3 methods are going to irreversibly change the protein so it can never do its normal function. But basically what happens is we can now eat it and consume it without getting sick. So for instance, when you apply pH or temperature or salinity to foods, what you're killing is the bacteria, the parasites, fungus that's on it. But what's happening is those guys can't do their normal function to make you sick. But what they can do is, because now they're denatured, we can take their proteins and make them our proteins. If we ate it raw, those things are still going to be alive, and they're going to really make us sick. That's why we change protein structures with pH, temperature, or salinity. There's 4 levels of how we make proteins, and this is a big deal because we're going to talk about this again later when we talk about genetic diseases, because typically these are the places where we're going to see mistakes, and the mistake is that we put the wrong amino acid in the wrong spot, and when we do that, that creates a protein that doesn't work anymore. Remember, structure equals function? Well, this is where that structure gets built. And if you mess it up anywhere here, the functionality is all wrong, and it won't work. So we're going to talk about these 4 levels of protein structure, and then later we're going to talk about it again and again when we get to genetic diseases. So this is the primary level, and you can see all the amino acids sort of stuck together with a certain shape. So they're basically a string, they're held together with peptide bonds. They're linear right now. What's going to happen is they're going to make these 2 shapes. Either this alpha helix, which is like this curly Q, or this beta-pleated sheets, which kind of looks like just a folded piece of paper. These are the 2 main shapes that they make. These guys are going to be hydrogen bonded. They're going to use these alpha or beta sheets, and that's going to complete their little shape. When the amino acid is completely filled, or when the protein is completely filled, you'll see that you could see that this is a combination of these curly Q's, which are those alpha helixes. See them right there? And then see these zigzag ones that are in pink? Those are those beta-pleated sheets. This could be a protein on its own, but more likely what's going to happen, this is called the tertiary structure. We're going to go to the quaternary structure, which is going to be you add protein 1, 2, 3, and 4, and together they come together as a functional protein. So that would be the tertiary, could be the functional protein, but more likely you add a bunch of them together in the right way, and they become the actual protein that's doing its work. If you don't put a protein correct, like the folding is messed up somehow, what happens is it loses its functionality. Remember, structure equals function? Well, this is what a red blood cell looks like. And this one that sort of looks like a little sausage, these guys are put together incorrectly. And because they're put together incorrectly, they don't work as well, or they may not work at all. These little things are called sickle cell anemia, and the round ones are the normal ones. So this is what hopefully we all have. And these ones that kind of look like maybe slugs, they're the red blood cells that somebody makes because somebody put in, when they were making the red blood cells, they put the wrong amino acid in. Their instructions are incorrect in one place. And because of that, this is the situation they have.